Namaste all of you. My name is Dr. Ritu Singh Rajput, Faculty of Agriculture and Veterinary Science, Jayoti Vidyapeeth Women's University, Jaipur. Today, in our session, we will discuss about high performance liquid chromatography, which is also called HPLC. HPLC, or commonly known as high performance liquid chromatography, is an analytical technique used to separate, identify, quantify each component in a mixture. This mixture is separated using the basic principle of common chromatography and then identified and quantified by the spectroscope. In the 1916s, the column chromatography LC and its low pressure suitable glasses columns are further developed to HPLC with its high pressure adapted metal column, and HPLC is basically high improved from the column liquid chromatography. Instead of solvent being allowed to the drip through a column under gravity force through the under high pressure up to 400 atmosphere. The purification takes place in the separation column between stationary and mobile phase. The stationary phase is granule material with very small pores particles in the separation column. The mobile phase, on the other hand, is a solvent or a solvent mixture, which is a force at high pressure through the separation column. Y valve with a connected sample loop is a small tube or capillary form the pump to the separation column using a syringe. Subsequently, the individual components of the sample migrate through the column at different rates because they are retained to varying degree by the interaction with the stationary phase. After leaving the column, the individual substances are detected by a suitable detector and passed on the signal to the HPLC software on the computer. At the end of the operation run chromatogram is the HPLC software on the computer is obtained. The chromatogram allows the identification and quantification of different substances. Instrumentation of the HPLC is, first of all, it is required pump, injector, column, detector, recorder, disgasser, and the column meter are its instruments which are required in HPLC. The pump, the development of HPLC lead to the development of pump system. The pump is positioned in the most upper stream of the liquid chromatography system and generate a flow effluent from the solvent reservoir into the system. High pressure generation is the standard requirement of the pump, besides which it should also be able to provide consistent pressure at condition and a controllable reproducible flow rate. Most pump uses current LC system generate the flow by back and forth motion of the motor driven piston. Because of its piston motion is produced pulses. An injector is a place the next to the pump. The simplest method is used as a syringe and the sample is introduced the flow fluids. The most widely used injection method is based on the sample loops. The use of auto sampler system is also widely used to allow repeatedly injections in a set schedule timing. The separation is performed inside the column. The recent column are often prepared in stainless steel housing instead of glass column. The packing material generally used in silica or polymer gel compared to the calcium carbonate. The eluting used to the LC varies from the acidic to basic solvents. Most common housing is made of stainless steel since stainless steel is tolerant towards a large variety of solvents. Detectors separation of analytes is performed inside the column where the detector is used to observe the obtained separation. The composition of eluting is the consists when no analyte is present. While the presence of analytes change the composition of the elutants, what detector does it to measure the difference? This difference is monitored as a form of the electronic signals. There are different types of detector available. The next is our recorder. The change is eluted detected by the detector is a form of the electronic signals, and thus it is still not visible to our eyes. In older days, the pen chart recorder was popularly used. Nowadays, a computer-based data processor is more common. There are various types of data processor, from the simple system consisting of the build, printer, and the word processor, while those with the software that are specifically designed for the LC system, which not only data acquisition, that features like sorry, fitting baseline correction, automatic concentration calculation, molecular weight determination. Together, the elutant used for the LC systems and lysis may contain gases such as oxygen that are non-visible to the eye. When gas is present in the elutant, this is detected as a noise caused an unsuitable baseline. Degasser use special polymer membrane tubing to the remove gases. The numerous very small pores of the surface of the polymer tubes allow to the air to go through while pretending any liquid to go through the pores. Column heater, in the order to the obtain repeatable system result, it is important to keep consistent temperature condition. Also for the some analysis such as sugar and the organic acid, better resolution can be obtained at evaluate temperature 50 to 80 degrees Celsius. Thus, column are generally kept inside the column oven column heater. There are some kind of HPLC we have discussed here. First is the normal phase. Column packing in polar, for example, silica, and the mobile phase is non-polar. It is used for water-sensitive compound, geometric isomers, trans isomer, and chiral compounds. Reverse phase, the column packaging is non-polar. The mobile phase is water plus miscible solvent. It can be used for polar, non-polar, ionizable, and ionic samples. Third one is our ion exchange. Column packaging contains ionic groups. Mobile phase is buffer. It is used to separate anions and cations. Fourth one is our size exclusion. Molecules diffuses into the pores of the pores medium are separated according to their relative size to the pore size. Large molecules elute first and smaller molecules elute later. Now, we will discuss about the applications. What are the application of HPLC? HPLC has developed into the universally applicable method so that it finds its use in almost all area of chemistry, biochemistry, and pharmacy. Analysis of drug, analysis of synthetic polymers, analysis of pollutants in environment analytes, determination of drug in biological matrix, isolation of valuable products, product purity, quality control of industrial products, fine chemicals, separation and purification of biopolymers such as enzyme or nucleic acid, water purification, pre-concentration of the trace component, ligand enzyme, exchange chromatography, ion exchange chromatography for proteins, high pH anion exchange chromatography of the carbohydrate and the oligosaccharides. Now we have talked about the advantage of high performance liquid chromatography. First is its speed. Second is its efficiency. Third is accuracy. And fourth one is versatile and extremely precise when it's come to identifying and quantifying chemical components. Now we will talk about the limitation. What are the limitation of HPLC? First is its cost. Flip side, it is an advantage. HPLC can be costly, requiring large quantities of expensive organics. Second one is its complexity. And third one is HPLC does not have low sensitivity for certain compounds, and some cannot be detected as they are irreversible absorbents. Volatile substances are better separated by the gas chromatography. So this is all about the HPLC. In our sessions, I will tell you the summary. First of all, I will tell you what is the HPLC. After that, the principle of HPLC. Then what instrumentations are required in HPLC, which is pump, injector, column, detector, recorder, degasser, and column heater. Then we will talk about the types of HPLC. They are normal phase, reverse phase, ion exchange, and size exclusion. After that, we have discussed the application of the high performance liquid chromatography, then advantage and the limitations. So this is the summary of HPLC. And as exam point of view, if they will ask any question about the, first of all, they will ask main question, what is the HPLC? And principle of the HPLC, you have to know about the principle mainly and the process and generally why we are using. So you have to know its advantage and its limitations. Thank you so much.